Hello students, a warm welcome to you in the virtual practical class on skin and in organs. In this class, we will see the different parts of the skin and we will identify the different cells of the skin. In the earlier classes, we have studied the luminal organs and how to identify the different luminal organs. But this class, we will see a structure which is present throughout the body, and as I have mentioned that if you can identify the lining epithelium, your 90% of identification of an organ is over. So in this class also I am telling identify the lining epithelium. So what are the competencies expected after learning uh, the skin and end organ histology? Of course, you will draw the diagram of skin, and that will help you to diagnose the different pathological conditions of the skin in the later years of life. Everybody will not be dermatologist, but basic knowledge of the skin is required for a doctor. The competencies expected are: you should name and identify the various layers of the skin, identify the various cell types of the skin, identify the sweat and sebaceous glands, know the structure of the hair follicle, know the various sensory end organs in the skin. So in our theory class we have learned about the different types of skin. The parts of the skin also we have learned. There are thick skin and thin skin. How to differentiate between the thick and thin skin? The thick skin present in the palm and the sole. This is also called glabrous skin. Thin skin present all over the body, and thin skin has hair follicles. The thick skin do not have hair follicles, but main difference is that please see that there is a amorphous thick layer, the keratin layer. These are the dead cells which forms a thick layer over the other layers of the underlying structures. So the presence of a thick layer of amorphous cells differentiates between the thick and thin skin. In the thin skin, if you see this, there is absence of this layer or keratinized layer. The keratinized layer is not absent in the thin skin, it is there, as it has been told in the theory that it is of few cell thickness, but you can see here because of the processing most of this keratin has gone, but there is few dead cells are there, and that form the stratum corneum of the thin skin. But the stratum corneum of the thick skin is very thick. The other parts of the skins or the parts of the skin are epidermis, the dermis and the hypodermis. In gross anatomy, hypodermis is equivalent to the superficial fascia, and there are elevations and depressions of the dermis and the epidermis projects into the dermis. So see this pink colored layer where there are multiple projections. This is the epidermis. Please identify the epidermis and dermis. The theory, it has been told what is the difference between the epidermis and dermis. So now you know difference between the thick skin and thin skin. It is the presence of the stratum corneum. If there is a thick stratum corneum, that is thick skin. If the stratum corneum is thin, that is thin skin. The different layers of the skin. I have taken a book picture and a picture from the slide available in our department. On the right side, if you can see, the first layer is the detaching squamous epithelium. See here also in the real slide you can see a detaching squamous epithelium. The next layer, or from where the detachment of the keratinized squama is occurring, is the stratum corneum or keratinized squama of the cornified layer. You can see the multiple layers of dead cells are there, where you can see only the margins of the cells. In between there are white substances. There is no nucleus. Here also in the drawing the nuclei has not been drawn. So in these cells nuclei are not there, and in case of the thick skin this layer may be up to 50 cell thickness. In case of the thin skin it may be up to 10 to 15 cell thickness. The next layer is the granular layer or stratum granulosum. In the schematic diagram, lot of granules has been drawn. These are the keratohyalin granules. Here also you can see the granular layer which contains keratohyalin granules. The keratohyalin granules has taken hematoxylin stain. So this layer is stratum granulosum and cell contains keratohyalin granules. The next layer is the prickle cell layer or stratum spinosum. Here also you can see stratum spinosum. We'll see great details in enlarged magnification of the same slide. There are lots of spine-like structure from the surface of these cells. That's why they are called stratum spinosum. There are the last layer is the stratum basal. The stratum basal contains darker cells. Here you can see, and their cuboidal cells. Their uh, nucleus is round and they are dark. So uh, at the base of the uh, dermal papilla one can see the stratum basal cells, and from these cells only the other layers develop. You can see sometimes mitotic figures. What are the other cells present in the stratum basal? The melanocytes. Earlier I have already mentioned the melanocytes are the dendritic cells. See here, lots of dendritic processes are there. The function of the melanocytes has already been mentioned, and the number of melanocytes in the white and the dark subjects are same. It is the melanin pigment, and the degranulation of the melanin determines the color of the skin. There is other type of dendritic cell is there. You can see there are dendrites here. These dendritic cells are the Langerhans cells. See the position of the dendritic cell. Here It is in the middle of the stratum corneum, but the melanocyte is present at the stratum basal. This is another type of dendritic cell, that is the Merkel cell or the sensory cell of the skin. It has a lot of processes. This is actually a neuron, and a nerve terminal has come and making synapses with it. So there are three types of dendritic cells are there in the epidermis: the melanocyte, the Langerhans cells, and the Merkel cell. This is a picture, high magnification picture of the available slides in our department. See the stratum corneum, the plasma membranes can be seen, and there is no nucleus. Stratum lucidum. In earlier slide, I have not mentioned about stratum lucidum. In theory lecture, I have mentioned what is stratum lucidum. This is a layer just above the stratum granulosum. The cells are not properly visible. It takes a eosinophil stain. Here, just above the granulocyte, there is a small streak of stratum lucidum. Stratum lucidum is absent in the thin skin. So, difference between thick and thin is keratinocyte or the stratum corneum layer, which is very thick in case of the thick skin, and presence of stratum lucidum in the thick skin. In the thin skin, stratum lucidum is absent. You can appreciate a eosinophilic layer just above the stratum granulosum. So this is the stratum lucidum. The next layer is the stratum granulosum, where you can see lot of keratohyalin granules in the cell. The stratum spinosum, those cells are very large, tile shaped, and in between you can see, in between the cells you can see the spine-like structures. Those are the attachment of the tonofilaments. Now the basal cell has not, stratum basal has not come, but certain other cells has been identified as melanocyte. The difference between the keratinocyte and melanocyte, see here, the melanocyte is darker, and there is a space around the melanocyte, because there is contraction of these cells. It creates a space around the cell. That is the only identification point of melanocyte in the light microscope. Sometimes dendritic processes can be seen. The Langerhans cells. The Langerhans cells are also dendritic cells, but the pigments are absent. They also have space around it, around the cell. The difference between the space around the cells in the keratin, in the stratum spinosum and the Langerhans cells, that see the attachment of the spines. In this space there is no spine, and see the position of these cells. These cells are present in the stratum spinosum. So that is how in the light microscopy melanocyte and Langerhans cells can be identified or differentiated. Here also I have taken a diagram from the Gray's Anatomy just to make you understand the slides. See the stratum corneum with the discomated cells. Then stratum lucidum is just above this dark color layer is the stratum lucidum, is eosinophilic layer. Stratum granulosum. There are 2 to 3 cell layers are there here. There are keratohyalin granules. Here also it has been drawn. Keratohyalin granules are there. So this is the stratum granulosum cell. Uh, stratum lucidum has not been drawn here. The stratum spinosum, this typical spiny appearance, has been shown here. That is the stratum spinosum. Here is the stratum spinosum. In between the cells there are spine-like structures. Then the Langerhans cells are there and the melanocytes are there. So here there is a melanocyte, there is space around these cells, and Langerhans cells I will show in next slide. This is a higher power picture of the thick skin. So in the left side you can see stratum corneum, the cells of the stratum corneum, and in between there are spaces which contains lipoprotein that protects the skin from, or it forms a waterproof protection of the skin. The stratum lucidum, see here the keratohynyl granules are there. This is stratum corneum, but just above it there is a red color area. So that is the stratum lucidum. Then stratum corneum already has been mentioned. Stratum spinosum, in higher power you can see the spines in between the cells. These are the stratum spinosum layer. This is stratum spinosum. Okay. Then the this is also stratum spinosum. The stratum basal already we have mentioned, but in the higher power magnification you can see certain cells with some space around and dark pigments are there. So dark pigments are the melanosomes inside it, and some of the cells here has taken up some dark pigments. These are the keratinocytes. Keratinocytes phagocyte the dendritic process, tips of the dendritic process, and it gives dark coloration. This is a slide of thick skin. Sometimes you may confuse it with a thin skin, but here the stratum corneum layer has been washed out, or due to some tissue processing artifact or bad tissue processing, the whole of the stratum corneum is washed out. But how you identify this as a thick skin? Because of the stratum lucidum. Stratum lucidum, earlier I have already told that this layer is eosinophilic, and it is a homogeneous layer. The keratohyalin granules are not there, or the nucleus is not there, and next to it is the stratum granulosum containing the cells with keratohyalin granules. They are this granules has taken hematoxylin stain. There are 2 or 3 cell thickness. Next to it is the stratum spinosum. Uh, in higher power you can see the spines. Now stratum basal, there are lots of cells with pigments. Here uh, the melanocytes can be seen. There is a cell here with some spaces around it. Here also another one. There is another one. This is another one. One can see the some of the dendritic processes also coming out, and the other cells which is visible, those are the stratum basal cells or the keratinocytes. They have engulfed the melanosomes of, or the tips of the dendrites of the melanocytes. So this is a skin from a dark colored person. You can differentiate between the epidermis and dermis here, but in the epidermis there are other cells also. See, this is a cell in the stratum spinosum, and there are a lot of spaces around it. So this is also a dendritic cell. This is Langerhans cells, and you cannot see typical spine which is there in the stratum in the stratum spinosum cells. So two types of dendritic cells you have to differentiate. The melanocytes are, see the position of the melanocyte. The melanocytes are at the stratum basal, and the Langerhans cells are in the stratum spinosum. Both the cells have space around it, and there is no junctional complex or the spines, but the Langerhans cells are present in the stratum spinosum. Melanocytes are never seen in the stratum spinosum. The dermis has two layers: the papillary layer, where the epidermal papilla are projected into it. So this layer is called the uh, papillary layer of dermis, and deep to it lies the reticular layer of dermis. The papillary layer contains thin uh, collagen fibers, and elastic fibers are also there. The reticular layer contains thicker collagen fibers. So that is the only difference. There is no demarcation between the two parts of the dermis. Now I will show you lots of slides to make you understand the skin. Okay. So from the dermis there are projections. Those are called the dermal ridges. They may be primary or secondary. So the whole thing is a dermal ridge from which the papilla are going, or dermal papilla is going inside the epidermis. So these are called secondary papilla or secondary ridge also. The function of this is to anchor the epidermis with the dermis. So this is the papillary layer of the dermis, which is projected in between the epidermal projection. So this is papillary layer, and deep to it is uh, the reticular layer. One can identify the thick stratum corneum, and up to the projections of the epidermal pegs. This is the epidermis, and deep to it is the dermis, and the projections of the epidermis, or sorry, the projections of the dermis into the epidermis, these are the papillary layer of the dermis. In the dermal papilla, one can see certain structures which has a lot of cells with flat nucleus. These are the Meissner's corpuscle. There are blood vessels also, one can see blood vessels, but it is not a typical section of a Meissner's corpuscle, but this is the position of this Meissner's corpuscle, that is in the papilla of the dermis. Already in the theory class, Meissner's corpuscle has been told in detail what kind of sensation it carries. Here is the uh, epidermis, the different layers of the epidermis. So one can see the stratum granulosum, above it is the stratum lucidum, stratum corneum. Here is the stratum basal, and the stratum spinosum is here in between or above the stratum basal. Here is the sweat gland and sweat duct. Sweat gland is a coiled tubular structure, so you expect uh, multiple lumens, and sweat duct is more or less straight, and they opens on the surface. So how to differentiate between sweat gland and the sweat duct? The sweat gland, actually it is also lined by stratified cuboidal epithelium, which is not seen here, but outside this you can see certain flat cells are there. They are the myoepithelial cells. In the sweat duct, these structures are lined by also the stratified cuboidal epithelium, but there is a difference in the lumen. The uh, sweat gland lumen is larger than the duct, and if you can see the color of the cells, the cytoplasm of these cells has taken darker stain ducts takes a darker stain, and uh, the gland cells takes a lighter stain. So the difference between duct and gland, see the lumen. Lumen is larger in case of the gland. The lining epithelial cells are lighter in case of the glands. Lining epithelial cells are darker in case of the sweat duct, and the lumen is smaller in case of the sweat duct. And there are myoepithelial cells at the base of the gland, which is absent in the sweat duct. The structure on the right side is also lined by stratified cuboidal epithelium with dark cytoplasm. So this is a slightly oblique or longitudinal section of the sweat duct. The other structure is also a sweat duct. See the lumen also here. Uh, the lumen is becoming narrower, so the uh, the gland is opening into the duct. So this kind of structure is very important to identify. Both glands and ducts are visible here. This kind of structure already you have seen. This is the structure of the epidermis. On the right side the stratum corneum is there, and the left side the other layers of the skin is there. There is stratum basal with certain cells, probably the nucleus around the space. This is a melanocyte, and uh, in the upper layer you can see there are spaces in between the cells with certain spine-like appearance. So this is the stratum spinosum. Again, this is a slide to show you the melanocyte and the Langerhans cells. The melanocytes are present at the stratum basal. So this box is enlarged here to show the melanocyte. Actually, typically you see this is the nucleus of the melanocyte, and the cytoplasm has not taken the stain, but there are periphery of the vacuole, there are uh, the dark substances are there. So these are the melanosomes in the melanocyte, and this is also another one. These two has already been shown, but the other cells here, what you see, this is not a melanosome, this is a keratinocyte which has engulfed the melanosomes. And in the upper part one can see the similar type of cell, a similar type of cell here which has space around it. So the cytoplasm has not taken the stain. This is the Langerhans cell. The function of the Langerhans cell has been mentioned. The left panel shows a schematic diagram of the skin with the hair follicle and the presence of the eccrine and the apocrine sweat glands. Theoretically, eccrine and apocrine sweat gland difference has already been told. Eccrine glands open directly on the surface. Apocrine sweat glands doesn't open directly. They open at the root of the hair follicle. There is another gland, that is the sebaceous gland, which also opens into the infundibulum of the hair follicle. And there is a muscle called the erector pylori muscle, which when it contracts, it helps the expulsion of the sebum into the hair follicle. And there are different layers of the hair follicle are there. There is internal root sheath, external root sheath, which is continuation of the epidermis, and there is a matrix. This matrix cells, actually here lies the stem cells, they divide to form the other layers of this hair. So here has a cuticle, here has a cortex and a medulla. The melanocytes are present at the matrix, they divide and they migrate in this layer. With aging, the melanocytes number decreases and also number of melanosomes decreases. So that makes the color of the skin gray in case of the old subjects. It is the melanocyte are absent or less in the matrix of the hair follicle. So that is the cause of graying of the hair, and it may happen uh, because of the other reasons also. If people become very stressed, the melanocyte also stops secretion of melanosomes, and that is the cause of premature graying of hair. Now come to the sebaceous gland. The sebaceous gland, as I mentioned, that sebum is a fatty substance. It doesn't take amatoxylin eosin stain, so it gives a washed out appearance. The whole of the gland is full of sebaceous cells, and these cells contain lipid secretions. The nucleus is present in the center, and in the periphery there are basal cells. Actually, the proliferation of the basal cell push the cells in the cavity or in the lumen of the gland, and these cells, the luminal cells, they die in the process of apoptosis. So secretion of the sebum is the cell debris uh, and the sebum. Uh, the cells are replaced by the basal cells. So when there is a contraction of the rectopili muscle, the sebum comes out. Sometimes this opening of the sebaceous glands are blocked, and there may be inflammation, and it causes putrefaction or pus formation or abscess formation in these glands near the hair follicle, that gives rise to acne. So acne is infectious or infections of the sebaceous gland. Here we have tried to take a section of the erector pylori muscle also. Some muscle fibers are visible. So this is the erector pylori muscle, and this is the root sheath. Actually, this is the hair follicle, or the terminal part or small part of the hair follicle is in here. These four pictures shows the sebaceous gland and a hair follicle. This is the root of the hair. This is the sebaceous gland about to open into the hair follicle, and certain muscles are there here. These are the erector pylori muscles. Here also there is another muscle, erector pylori muscles. Here is a transverse section of the hair follicle of the sebaceous gland, and the base of the hair follicle is there. Here is some muscle fibers are there. These are the erector pylori muscles. Here also is the sebaceous gland and the terminal part of the hair follicle with some muscles, the erector pylori muscles. This sections has already been shown you. Just to show you the stratum basal containing highly colored or highly pigmented epithelium, melanocytes and keratinocytes. Thick skin. See the stratum corneum layer, then stratum granulosum and the stratum spinosum, the basal cell layer. Already it has been explained. This is a thin skin with the hair follicle. Upper panel in lower magnification. This is also in higher magnification of a hair follicle and the sebaceous gland. Muscle fibers are there. This is actually typical erector pylori muscle. The epidermis is going inside to form the outer root sheath of a hair follicle. The sebaceous gland at the base of the hair follicle. Here also uh, the hair follicle. This is a transverse section of hair follicle, and there are multiple transverse sections are there in this hair follicle. I think we should end now with the note that this is a section of skin. This is a thin skin because you see the stratum corneum has few layers of cells, and there is no stratum lucidum. This is the whole thing is epidermis. The lower part is the dermis. Here in the thin skin it is showing the hair follicle, although the sebaceous gland is absent. Maybe it is in the other plane, it has not come. Here also you can see the beginning of a hair follicle. So thank you all for patience hearing, and I think this will give a clear picture of the skin and end organs. I am not able to show you the Pacinian corpuscle because in these slides there is no Pacinian corpuscle. In the theory class it has been shown the Pacinian corpuscles. Okay, coming back again to show the position of the Meissner's corpuscle. Actually, in between the epidermal projections, this is the dermal papillae, and in this layer you see there are certain elongated structures. These are the typical Meissner's corpuscle. So the nerve is in the center. Actually, the nerve is it takes a helical way or zigzag structure, and around the nerve there are certain cells covering the nerves. So it gives a typical appearance of the Meissner's corpuscle. Sometimes it is sectioned obliquely, so you cannot see a typical picture. This is a typical picture of a Meissner's corpuscle. This is actually trichrome stained section, so it is showing some different colors also. Thank you all. Welcome back. I forgot to mention the difference between the eccrine and apocrine sweat gland. See, there are 4 slides here. The upper panel shows the apocrine sweat gland. The lumen of the apocrine sweat glands are larger than the eccrine sweat glands. Both the apocrine and eccrine sweat glands are lined by stratified cuboidal epithelium, but see the lumen. The lumen of these glands, the apocrine sweat glands, are larger than the lumen of the eccrine sweat glands. The other differences are the uh, the glandular epithelium are of one type in case of the apocrine gland, and here the glandular epithelium are of 2 types. It has dark cells and clear cells or the light cells. So the apocrine sweat gland uh, structures, one can see the myoepithelial cells are there also. So here are the presence of myoepithelial cells containing elongated nucleus, In both the cases, myoepithelial cells are there. So main difference is the lumen and the epithelial lining. The epithelial lining is single type of cell, that is the and here there are dark and light cells. The mechanism of secretion in case of apocrine is the apocrine the merocrine type, and eccrine gland has a different mechanism of secretion. Thank you all.